Hello, everyone, and welcome to today's webinar, Bench to Clinic, Preparing for a Successful Transition, presented by Roe. I'm Joe Keenan, contributing editor to Fierce Markets, and I'll be your moderator. Our speaker today is Dana Minnick, Senior Research Scientist at Roe. You can read her full bio on the right side of your window. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the Resources List button at the bottom of your screen. The webcast is being streamed through your computer, so there's no dial-in number. For the best audio quality, please make sure your volume is up. You can find additional answers to some common technical issues located in the Help button at the bottom of your screen. This webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentation with a Q&A session. Please submit your questions using the Q&A button on the left side of your screen. That pretty much covers the technicals. I think we're ready to begin. Dana, please go ahead.
Thank you, Joe. Okay, so due to some technical difficulties today, I'm going to have to say next slide to advanced slides for today. Our discussion today is really going to focus on planning and preparing for a successful IND application, and this will entail covering the disciplines clinical, non-clinical, and CMC, as well as regulatory, including pre-IND meetings and IND. So it's a lot of topics to cover. It's going to be somewhat high level, but hopefully there'll be some take-home key messages that will be helpful for you as you move your program through from the bench to the clinic. So next slide, please. Shown here is some of the barriers that need to be overcome when you're moving your research program through preclinical development from the bench and into the clinic through a successful IND and into a clinic. And that includes funding, of course, and then pulling together the right translational team, having intellectual property, and having the appropriate regulatory knowledge in your team. Next slide, please. Putting together an integrated development program is really essential to map this out at the beginning of your once you have a candidate selected, and even before that. It's important to begin with the end in mind, and that includes pulling together the appropriate interdisciplinary project team, which includes clinical, non-clinical, CMC, and regulatory experts. These can be in-house or they can be external consultants. So you may also want to involve labeling and marketing teams or consultants to help you understand the marketing demands and expectations for your product, the competitive landscape, and the implications of your clinical program on the marketability of your product. A strategic development plan should outline at a minimum your product's indication, target patient population, the route of administration, and primary endpoints that you plan to evaluate in your proposed clinical studies. The development plan is a living document that should be revisited and revised throughout the clinical program, obtaining input from your interdisciplinary team and agreement on any new or modified strategies that you'll be pursuing. It is important, where possible, to introduce some go, no-go rationale at specific points during your program, at which point you determine to either proceed with or terminate your current development strategy based on some predetermined criteria. Next slide, please. So one strategic tool that can be used to help map out your development program is a target product profile. The target product profile, also known as TPP, is a living document as well, and it summarizes your drug development program in terms of labeling concepts. The target product profile should describe the overall intent and goals of your product development program. It should summarize current and future studies or existing data, possibly from the literature, that can be used to justify the intended label claims, and to outline any additional studies that may be needed to support your program. And also to identify any risks that might be associated with your development strategy that will need to be addressed or mitigated during development. The TPP should also include information on the competitive landscape for the product and key differentiators between your product and the current competition on the market, making sure to include a comparison to the gold standard for your product. For example, oftentimes you'll have your desired criteria for your in your TPP, and then your minimum acceptable criteria, and then and ways that you'll be differentiating yourself from the gold standard. You may want to revisit your TPP throughout your development program. It's a living document, particularly following major milestones and decisions in your program. So in addition to being a helpful tool, the TPP can also serve as the basis for your annotated package insert or your label that you'll be submitting in your marketing application. So it does really benefit you to get started on this early in your product development. So on the next slide, I have an example of a target product profile. This was taken directly from the FDA guidance for industry on target product profiles. As I mentioned, the TPP should outline the proposed target label language for your product, as well as specific studies or evidence that in the literature that would support your intended labeling claims. The guidance includes an example milestone table here, as you see, to indicate when and if the TPP has been submitted to the agency for review. It can be submitted as part of your briefing document during development meetings, or it can be submitted separately to the FDA for their review and input. On the next slide, just a slide depicting the integrated product development plan, beginning with the end in mind, thinking about labeling claims for efficacy and safety and what you want on your package insert, your label. From there, you'll design your phase 3 studies to support your intended label claims. Your phase 1 and 2 studies will be designed to support your phase 3 studies. And then your non-clinical toxicology and chemistry manufacturing and controls programs will be designed to support your proposed clinical program and then marketing authorization and commercialization. So the idea is to plan backwards and execute forward. And the next slide is a slide really that summarizes what I just described, and that is that really you're going to begin with your phase 3, and then you're going to work backwards. It is really important to consider the potential need to investigate your therapeutic agent in special populations and or the potential for drug-drug interaction studies. Also, immunogenicity testing may be needed if you're working with a biologic. And that requires a lot of upstream activities in terms of method development, validation, et cetera. And so the timing of when you need to have certain studies done along a timeline is really critical. And then on the next slide, I depict this. Here is a preclinical development from candidate selection on the left side, all the way through to an IND submission on the right side. And the colors are kind of somewhat by discipline, not exactly. The important point here is that there's some flexibility in when you decide to run your toxicology studies during your preclinical development program. Shown here, they have the gene tox studies run earlier because for this particular program, that was a go-no-go criteria for them. They could not tolerate a genotoxic molecule. So, however, one of the important caveats is that you need to have your active pharmaceutical ingredient made to support your tox studies, and this is actually a fairly considerable amount that needs to be made, and it needs to be made in the same processes as the drug substance that you're using for your clinical trials. All of your impurities in your clinical trials need to be covered in your non-clinical tox study lots. And then there's some analytical method development and validation and bioanalytical method and development and validation that needs to precede the conducting tox studies. On the next slide, the non-clinical development plan is designed to provide a comprehensive assessment of the pharmacology, pharmacokinetics, and toxicity of the drug substance and product. The non-clinical development plan supports the clinical development plan, CMC activities, and marketing authorization. The data can also be used to specifically for dose selection for your initial clinical trials, for pharmacodynamics and biomarker development for early phase clinical trials, to understand a therapeutic index, either overall or for specific toxicities that are seen non-clinically, and to inform specific safety monitoring or use in specific populations. And a lot of the non-clinical studies will be used to support the label, and they'll be directly genetic toxicology, reproductive toxicology, carcinogenicity studies. They'll be directly supporting the label. On the next slide, I just outlined a number of the non-clinical regulatory requirements for initial IND according to ICH M3 for a small molecule or S6 for a biologic. For general toxicology studies, safety pharmacology studies are expected. For small molecules and peptides, genetic toxicology studies are required. Toxicokinetic studies are also required to understand the pharmacokinetics at the doses that were used in your tox studies. And in vitro drug-drug interaction studies may be needed if your initial studies are in patients on concurrent medications. And those would include cytochrome P450 inhibition and induction studies, as well as BCRP and PGP transporter studies. These are in vitro studies, and typically they can be done concurrent with your phase 1 program, but oftentimes if you're going to go early into your patients, you'll want to have those done prior to your IND. On the next slide is just a little bit more deeper dive into the pharmacology and pharmacokinetic studies that are expected in an initial IND. Primary pharmacology studies are expected, and these could include potency assays. Either they're usually in vitro assays, either cell-based or enzyme-based assays, and they can be a potency of your molecule against the target, either the human target and oftentimes in comparison with your tox species targets. So you can understand the relative potencies of your molecule in your tox species versus humans and the human target. And secondary and safety pharmacology studies are also often required. These assess off-target activities. Safety pharmacology focuses on organ systems. The core battery is typically expected for an initial IND. There may be some additional organ systems that need to be evaluated depending upon your class and any findings that you may have in your tox studies. CNS, respiratory, and cardiovascular make up that core. So PK studies are expected, toxicokinetic studies are expected to be conducted to support your general toxicology studies. If you have a therapeutic that's given in a repeated dose manner, you'll want to have single dose and repeat dose toxicokinetics, so you have an opportunity to look to see if there's any change after a single dose versus after repeated doses, such as accumulation, and whether or not there's any sex differences, and if the toxicokinetics are dose proportional. Metabolism studies, in vitro metabolism studies in hepatocytes or human liver microsomes are also very valuable to have prior to your IND. These studies would look at the metabolites in, say, human hepatocytes versus the metabolites in your tox species and maybe even your pharmacology species hepatocytes. And it's important to note that all of your human metabolites need to be covered in your tox species. And if they aren't, then that's probably an opportunity to have a conversation with the FDA at a pre-IND meeting. And as I mentioned before, potential for drug-drug interactions. If you're going right into patients, you'll want to assess that. On the next slide, genetic toxicology studies are expected for small molecules and peptides prior to phase 1. The in vitro studies, the bacterial reverse mutation assay and clastogenicity assay in mammalian cells are expected for even a single dose in humans. The in vivo genotox assay is needed before repeat dose studies in humans, although oftentimes people will, sponsors will conduct these studies prior to the initial IND. On the next slide is just some information about general toxicology studies. These studies are typically conducted in rodents and non-rodents. However, they need to be conducted in relevant species, and oftentimes for biologics, that's a single species such as a cynomolgus monkey. They need to be conducted for the United States for FDA. 1 day of tox, it covers 1 day in the clinic. 14 days of tox covers 14 days in the clinic. It's a 1-to-1. For Europe and other countries, that maybe 14 days may be required as a minimum. The purpose of the general toxicology studies is to determine a safe dose and to identify target organs of toxicity. And then the toxicokinetics and/or exposures that go along with safe dose and with certain toxicities that might be seen. If you're working with a biologic, you want to assess immunogenicity in your general tox studies, the potential for immunogenicity in your general tox studies. And this is important not because it does not inform whether or not you're going to have anti-drug antibodies in your human studies. It's not predictive, but it does help you to interpret the results that you'll get from your general tox studies. Dose range finding studies are often conducted prior to your pivotal tox studies. Very rare that wouldn't happen, and it's really important to pick the right doses for your general tox studies. For chronic indications, you need up to 6 months of dosing in a rat and up to 9 months of dosing in a non-rodent prior to conducting typically your phase 3 clinical trials, and certainly for marketing authorization. On the next slide, dose selection, as I mentioned, for general tox studies is really important. It's really critical. For small molecules, it's different than for biologics. For small molecules, the low dose, similar to the anticipated clinical exposure, could be a small multiple above that. The high dose needs to meet one of the 4 following criteria. It needs to be either the maximum tolerated dose, the maximum feasible dose, at least 50-fold above the anticipated maximum clinical exposure. And if you choose that one, you may be limited to how high you can dose in the clinic. You may be limited to 1/10 of that exposure. And the limit, or the human equivalent dose, or a limit dose, which is 1,000 mg per kg if you have a human dose of less than 1 gram per day, otherwise 2,000 mg per kg. And you'll want to choose at least 3 doses for your general tox studies. For biologics, because oftentimes biologics, you don't have any obvious MTD or a obvious severe toxicities or toxicity profiles. So the low dose is typically your minimum biological effect level, also called MABEL, and your high dose is oftentimes typically at least 10-fold above your anticipated maximum clinical exposure. Sometimes that's not feasible because of the concentration of your protein in the formulation or other reasons. And if you have a small margin that for your maximum feasible dose versus where you need to be in the clinic, that's a good conversation to have with the FDA at a pre-IND meeting. On the next slide, this depicts a therapeutic index and what that means, and that would mean really a comparison of the minimum efficacious dose or exposure in your human clinical trials, and you compare that with your NOEL and your most sensitive species, or your the dose or exposure at your NOEL. And the therapeutic index, what's a reasonable therapeutic index, really depends upon the target indication, the severity of the disease, and whether or not there are other therapies available for that indication. On the next slide, dose selection for phase 1 studies can be based either entirely on your non-clinical safety studies, or they can be based in part on that, and in part on an anticipated efficacious dose in humans, whichever one is lower. There's a guidance estimating the maximum human starting dose in normal healthy volunteers. It's an FDA guidance, and they recommend a starting dose of 1/10 the human equivalent dose of the NOEL in the most sensitive toxicology species. This is just a general guide. If you have a severe toxicity, a steep dose toxicity curve, an unmonitorable or a toxicity that isn't reversible, then you may want to have an increased safety margin for your initial dose. Dose escalation is typically in increments of 2 to 3-fold. It really depends upon your therapeutic area and what you know about your class of molecules. And that would also depend upon a safety concern. You may want to decrease those increments if you have significant safety concerns. You may want to do some safety evaluation in between dose escalations. The maximum dose in a clinical program typically does not exceed the human equivalent dose or exposure for your NOEL in your tox studies, or 1/10 of your NOEL exposure. But that is also can be changed depending upon, can be exceeded depending upon data on a case-by-case basis. The next slide depicts examples of how you might incorporate some of your preclinical testing results in your clinical program. And 2 examples here. One is QT prolongation. You have QT prolongation. You see that in your hERG in vitro human ether go hERG study, and also maybe in your cardiovascular in vivo studies. You might want to incorporate more ECGs into your initial clinical trials, and possibly even some Holter monitoring, depending on what you find in your tox studies. If you have increased liver enzymes or liver toxicity, you will probably want to exercise caution if you're going into patients who may be on other medications that may also have liver effects, and you'll also likely want to have some additional liver monitoring in your clinical trial. On the next slide is we're switching gears now to CMC, chemistry, manufacturing, and controls. And the amount of information that you put in your initial IND really needs to be sufficient so that the agency can be assured that your therapeutic agent is properly identified, that there's quality, purity, and strength is understood. And that's the active pharmaceutical ingredient, the API, the drug substance, the amount that you're going to need for your tox studies and for your initial clinical trials really needs to be calculated at various stages in the development program, because those are gating oftentimes to being able to run the studies. And it's actually pretty astounding how much API you need to run tox studies. And precursors to even making the active pharmaceutical ingredient, you have to set up your assays, your analytical assays, and at this point in the development of preclinical, they need to be qualified, not really validated per se. Pre-formulation and formulation development needs to occur prior to your submission of your IND. Where that occurs depends upon what the route of administration is, potentially. If you're working with a topical or an ocular route of administration, you'll want to use your formulated material for your clinical trials in your tox studies. And so therefore your pre-formulation and formulation development needs to move earlier in your program. And you'll need to have a certain amount of stability prior to when you submit your IND. And that can be either from engineering batches made in the same process or directly from your GMP clinical batch. Okay, so switching to the next slide, now you've gotten through some of your tox work and your non-clinical work, and you have initiated some of your CMC activities, and you have a protocol synopsis, and you've thought about your clinical development plan. You have that mapped out. It might be time to consider a pre-IND meeting. And why would you want to have a pre-IND meeting? Really is to focus on scientific and regulatory issues. I list several of them here, including your clinical trial design. If you have any toxicity concerns or unique metabolites, this is a good time to discuss them. If you have a novel formulation or you have dosing limitations in your tox studies, this is also a good time to discuss them. If you want to discuss your plans for assessing immunogenicity in your clinical trials, this is also appropriate time to discuss that as well. And then there is a lot of data that and studies that demonstrate that having a pre-IND meeting really does increase the probability of a successful IND. The next slide talks about the timing of the pre-IND meeting. It's really important to note that the FDA is not a product development consulting firm. It's really important to get help from industry experts when you need it. And really, you can think about your pre-IND briefing document, your meeting package, as really the beginnings of assembling your IND, such that a lot of that may be able to be completely repurposed into your IND. And oftentimes, a pre-IND meeting would occur 2 to 3 months prior to the planned IND submission, at least that time. But really, you just need to have adequate information so that you can get your answers and have the dialogue and get advice from FDA, get their input. If you have a novel therapeutic or with a very novel therapeutic, you may want to have your pre-IND meeting earlier. The meeting request letter is sent out about at least 60 days prior to requesting, prior to your formal meeting when you plan to have that Type B meeting. And the meeting request letter, there are several elements that are required to be in this meeting request letter. There's a guidance that delineates those. Some of them include specific objectives and expected outcomes of the meeting, a proposed agenda, a list of questions, who the sponsor and attendees are going to be, and requested FDA staff, and some suggested meeting dates and times. And as I said, that needs to be submitted at least 40 days prior to the anticipated pre-IND meeting. And the next slide just shows what sort of information you may want to consider and get agreement on in your pre-IND meeting. You may want to lay out your high-level development plan through marketing. This is possibly a good time also to share your target product profile of what you have at this point. You may or may not have completed your IND-enabling toxicology studies. Oftentimes, if your therapeutic area is in your class is precedented, you may not really need to have your pre-IND meeting until after you've completed your IND-enabling studies, and then the meeting's focused entirely on your clinical plan and your CMC. CMC oftentimes is still a work in progress at the time that you're having your pre-IND meeting, but it's a good time to get confirmation on your drug substance and drug product release and stability specs that you're proposing for your early development, and any necessary development activities and studies that you're going to be doing. And of course, it is important to have at least a phase 1 protocol synopsis included in that meeting package to get some agreement with FDA on your proposed plans. On the next slide, the meeting information package needs to be submitted at least 30 days prior to the meeting. It should. There are some also required elements in this meeting package that are available in the FDA guidance on formal meetings. Typically, if you're sending hard copies of your meeting package, and you're going to send them in 3 hard copies to the document room at FDA, the regulatory project manager will also request oftentimes additional dust copies for the reviewers, and oftentimes I'll just throw a disk or a thumb drive with the same information on it for the RPM. They oftentimes appreciate that. On the next slide, just some ideas to think about for getting ready for your pre-IND meeting. I think it's really important to consider playing devil's advocate here and brainstorming possible FDA questions and answers. You can think about what questions that you've listed and information are going to be ones that need to be prioritized for a discussion at the meeting. And depending on how much experience you have with FDA meetings, you may want to have some mock meetings, some rehearsals. And if you have your FDA preliminary responses back, that's a really good time. On the next slide, FDA pre-IND meeting responses typically come in a few hours to a few days prior to the meeting. FDA meets about a week prior to the meeting. They meet internally, and then they generate preliminary responses, and then those are sent off to the client. And like I said, usually a few hours to a few days prior to the meeting. So it's really important when you get those preliminary responses to review them thoroughly and identify and prioritize questions and issues for discussion at the meeting. At that point, you can also decide whether or not you want to continue with the meeting, or should you cancel it because all the responses that you've gotten from the FDA are acceptable. That's more rare in my experience. On the next slide, just a few tips on the meeting day. It's important to arrive at least 30 minutes early, approximately 30 minutes early. You do need to go through security. If you are a foreign national, your paperwork needs to be submitted at least 2 weeks prior to the meeting. It's really important to keep your introduction when you're in the room. The FDA will be on one side of the room, and the sponsors, attendees will be on the other side of the table. And it is important to keep your introductions short so that you can spend the bulk of the time on the questions and answers. It is really important to be mindful of the time that you're spending on the question. If it seems like you're not getting anywhere, it might be possible to say, well, let's take this, let's just table this for now, and we'll come back to it if there's time, or maybe we'll follow up with an additional discussion at another time. It is also really important that the team know who is responsible for what questions and discussion, and also what things they're allowed to commit to or not commit to. It's really important that the team know that they don't need to make a commitment in the meeting. They can thank the FDA for their advice and say they'll take it back to the team for consideration. On the next slide, just denote some post-meeting activities. Hopefully you've had several people in your meeting taking their own good minutes for the meeting. You'll want to draft those as soon as possible after the meeting, and you can send those to your regulatory project manager within a week at most. They may use those in part to draft their own minutes. Although oftentimes I see now some of the regulatory project managers will have the minutes projected up during the meeting, and they'll be drafting the minutes during the meeting so that they'll get the sponsors buy-in to the meeting minutes right away. You'll receive the official minutes within 30 days of your meeting, and it is also important to understand that the meeting agreements are not binding. They may hold you to them, but they may also change their mind. And here's a case study. In this case study, a pre-IND meeting, the next slides, a novel small molecule oral drug is being developed for a rare disease, and the pharmacology studies support the proposed indication. The initial dose range finding tox studies suggested a steep dose toxicity curve with severe toxicity observed in rats. GLP tox studies were conducted and well tolerated in dogs. There was some liver toxicity at the highest dose. However, in rats, the NOEL was very low, in 1 mg per kg, providing a very small safety margin. In the pre-IND meeting, one of the questions, as a typical non-clinical question, is we've conducted all these studies, and does the agency agree that the preclinical development program, the list of studies, is sufficient to support the proposed clinical plan? And so sometimes the FDA won't really be looking at the actual data that you have in your package prior to answering this question. They'll say, well, yeah, the program appears to be adequate to support the proposed clinical phase 1 clinical study, but a full assessment will be made upon review of the audited full study reports. So that's on the next slide. and that will be included in the submission. And so on the next slide, the clinical question would be maybe the toxicities that were observed in the rat studies are monitorable in humans prior to the onset of severe toxicity, and we have a reasonable safety margin, potentially, or small, but reasonable, and they're manageable. Does the agency agree with the proposed start dose and the proposed dose escalation scheme? And one of the outcomes from that may be the FDA written response in this case was no. Given the severe, although manageable toxicities, this is on the next slide, that were observed in rats, the start dose should be less than 1/120th of the human equivalent dose of the NOEL. So they wanted to take a bigger safety margin. And the maximum exposure, they're also saying, should not exceed X amount. They wanted to cap the exposure in humans. And they also want a review before dose escalation, review of the safety and pharmacokinetic data from each cohort. So an outcome of this pre-IND meeting was that the sponsor agreed to start the clinical trial at a low dose, and that dose escalation would, with careful review and monitoring, would be conducted by an external data safety monitoring board. They also added some clearly defined stopping rules and some additional eligibility criteria. So on the next slide, I think we're move to talking about now the IND application. You've conducted your tox studies, you have your protocol, you've gotten your input on your protocol from your FDA, you have manufactured your drug substance and drug product for your initial clinical trial, and you're putting together your IND application. And so what information needs to be included in the IND? And this is from the Code of Federal Regulations. The focus is on the protocol and the general investigational plan. Although there's all the other supporting data is required, and it needs to be there. On the next slide, just a schematic of the format of the common technical document that is used typically to submit an IND or an IMPD. And here we see there are 5 modules. Module 1 contains regional information. It'll also contain your investigator brochure and your general investigational plan. Module 2 will contain all your written summaries for quality, non-clinical, and clinical. And then the bulk of the information and data are contained in modules 3, 4, and 5, the quality, the non-clinical reports, and the clinical reports. On the next slide, just a schematic about, in the U.S., original IND clearance and sort of the pathways here. So you submit your IND to the FDA, and there's a 30-day review period. At the same time you submit that, you may be also submitting your protocol and your informed consent form to your local or central IRB for their review. And then if you have a single positive opinion on your protocol and your ICF, as well as you haven't heard anything and 30 days have gone by since FDA has received your IND, you may begin your phase 1 clinical trial or your initial study. If you have any deficiencies, you may get a phone call during that 30-day review period by the FDA. Some of those deficiencies can be actually remediated during that review time, so it is really important to be available and because they'll expect very quick turnaround. There are some deficiencies, however, that can't be addressed during that time, and that's when there's a potential for a clinical hold. On the next slide, just some key considerations as you put together your IND application for your initial IND. And are adverse effects and toxicities, are they adequately addressed in the IND? Are the risks and benefits of the drugs adequately discussed and considered? Does the information provided support an acceptable margin of safety? And is the proposed clinical study design justified by the supporting non-clinical data provided in the IND? On the next slide, I've outlined just bulleted a number of reasons that I have seen for clinical holds. One of them often see is unqualified impurities in your clinical batch. They aren't qualified. They either weren't present in your tox batch, or they weren't present at sufficient amounts in your tox batch given your NOEL. to be considered qualified. And that's really important to really look at that before you move forward with your IND. You may have a poorly characterized cell bank or a viral bank. Your maximum dose may not be supported by the non-clinical data. That oftentimes changes. Clinical protocol changes are oftentimes things that may be able to be remediated during that 30-day review period and may not actually result in a clinical hold. However, if you have inadequate information from a non-clinical standpoint to evaluate safety, such as you didn't do any additional investigative work for a particular toxicity that would be concerning, that could also be a reason for a clinical hold. And on the next slide, we have another case study. And on the next slide, this is a case study of a 30-day safety review. And in this case, an IND was put on hold. During this 30-day safety review period, there were two main hold issues prior to allowing the first study in humans to proceed. One was a lower starting dose, and the other one was additional preclinical testing to characterize the toxicity noted in the completed preclinical studies. And so I think there's a few lessons learned there. They're bulleted here, but it really is important to actually consider whether or not you have an adequate safety margin and a justifiable calculation for your safe starting dose. And have you sufficiently characterized the toxicities that you may see in your preclinical program if they're warranted, such as if they're irreversible and or if they are unmonitorable, etc. Or you have a very steep dose toxicity curve. There's a few reasons why you might need to do some additional work. And the next slide is another 30-day safety review case study, and this one is during the 30-day safety review period, the FDA requested to have a conversation with the sponsor to discuss unqualified impurities noted in their GMP batch, and they wanted to discuss new impurities present in the clinical batch that were above the qualification threshold, and then the level of impurities present in the clinical batch relative to the non-clinical batches for those that exceed the qualification threshold, which is there's an ICH Q3A and B that determines, that shows the levels for qualification. And then they also want to discuss non-clinical studies that would be needed to qualify these impurities if they couldn't be qualified based on literature, which often is the case that they can't be. And so the lesson learned there, I think, is that it really is important to look at your impurity profile to make sure that your impurity profile for your clinical batch is qualified by your non-clinical tox studies in the batches that were used there. And on the last slide I have here for today, just some important factors for having a successful IND, and that's start your drug development program thinking about the end product. What do you want on your label? And plan the timing of your pre-IND meeting so that sufficient data is available to inform decisions. Design your clinical plan to be adequately supported by the non-clinical data, and build a strong relationship with the FDA. And last but not least, be forthcoming with potential issues of concern. They do usually come out during or after a regulatory review, and they can impact credibility. So it really is important to sort of air your dirty laundry, so to speak. So that is all I have for my presentation, and so I'm happy to answer any questions.
Dana, thank you. So why don't we move on to the Q&A. Just a reminder to the audience, there's still time to submit questions using the Q&A button at the bottom of your screen. We already have a lot of great questions, and we'll try to get to as many as possible. First up from the audience is, we are developing an already approved drug for another indication using a novel formulation. Can we rely solely on the agency's finding of safety and efficacy, or do we need to conduct any additional safety studies?
Okay. So for novel formulations, they actually require their own safety package. According, and there's an FDA guidance that will describe that. studies will be needed, and actually it depends really on the duration and the route of administration. So if you have shorter duration, you may need fewer studies. Typically you'll need gene tox studies, you'll need general tox studies, and you'll need maybe some reprotox studies at some point in your development plan. And you'll need longer chronic study, more chronic studies if you are intending for a chronic use. So novel formulations, even though you have an approved drug, that novel formulation will require its own safety package.
Another in the audience says they have a drug in development for one indication, but they also want to investigate the drug for another indication. Do they need to open a new IND?
In most instances, yes. If not, it's on a case-by-case basis. A new IND is typically required. However, this IND can cross-reference any applicable non-clinical CMC and clinical information from the original, the IND for the other indications, so that it shouldn't be, what would be new would be any new studies that would be needed for that new indication, if there were any, and your clinical protocol and your investigator brochure and your general investigational plan. So it could be streamlined, but typically, yes, a new IND is required.
So another person in the audience says their company is developing a therapeutic for a new indication and a new route of administration in the U.S. The drug is marketed in Europe and many other countries, but not in the U.S. Can they rely on EMA's or CA's findings of safety and efficacy for the non-clinical studies and some of the clinical studies?
Thanks. That's a good question. I think a lot of people think that they can, but unfortunately FDA will not accept another agency's finding of safety and tolerability to support an IND or an NDA. However, you can look at and do a comprehensive summary of the available safety literature and potentially some clinical pharmacology literature, and you may be able to defer and or waive some studies if you have a good scientific rationale and with safety and safety concern, that safety is taken care of. There needs to be adequate assurance of safety. New amount of administration, new indication and formulation also further necessitates additional studies. There will be some studies you'll have to do, unfortunately.
Okay, this is kind of a three-part question, and so it's a little long-winded, but I'll try and get through it. This company already has a pre-IND meeting for their therapeutic prior to conducting a pivotal toxicology study. It says, we have recently obtained results from these studies and have some questions with respect to how the toxicology results may inform the proposed clinical program. Can I request another pre-IND meeting? If not, what are my options?
FDA grants one formal Type B pre-IND meeting. In most instances, they hold to that. I've only seen one instance where they didn't, and that was when the therapeutic agent, the drug, was licensed to another company in between the original pre-IND meeting and the IND. So yes, one pre-IND meeting is all you have. So it's really important to time your pre-IND meeting so that you have sufficient information to get the information that you need from FDA. Sometimes that's not possible, and you have things come up later after your pre-IND meeting. What are your options? You could have a Type C meeting. The timelines are longer for that. That's 75 days versus 60 days from the meeting request letter. You can also request a written response, but they may still be on the Type C meeting timeline. Thirdly, I guess you could communicate with the regulatory project manager to discuss what they might think are the best, potentially the best options for you. And again, I just iterate that the timing of the pre-IND meeting, when you want to have that in your program, really needs to be thought out well, considered.
I kind of touched on this a little bit, but I think it's got a different twist to it. Is the IND process the same for additional indications of the same compound, or is there a simplified process if an initial IND has already been accepted?
It is. So this is essentially the similar question that I've already answered before. You can cross-reference your initial IND if you have a new indication. So you can cross-reference as much information as you want, and so those, that's a very simple process. You'll send a letter to the FDA allowing cross-reference from the initial IND to the new indication, and especially you'll need that if it's a different sponsor that holds the initial IND. And then you'll put in also your cross-reference in the various sections of the IND, and then you'll provide the letter of cross-reference also in your IND for your new indication. But it's streamlined, but you still need to have at least some of the parts. And like I said, the protocol, the investigator brochure, general investigational plan, those are all probably new.
What is the minimal duration of GLP tox for drug candidates with repeated administration in order to support IND and phase 1?
Okay, well, in the U.S., it's really a 1-to-1. So if you want to do a single study first-in-human study, a single dose, then you need a single dose GLP tox study, and typically in rodents and non-rodents. And if you want to have a 2 weeks multiple ascending dose study in humans following your single ascending dose in humans, then you'll want to make sure that you have at least that 14-day coverage in your tox studies. Oftentimes people will just do a 28-day study because the cost isn't all that much different, and they get a lot more coverage in terms of how much they can dose in the clinic. And then you can go either from those 14 or 28-day studies to a 3-month study as you increase the duration of your clinical trials and your phase 2 studies. And then ultimately, for a chronic indication, you'll need to have 6 months in the rat and 9 months in rodents and 9 months in the non-rodent studies to support typically your pivotal phase 3 trials and marketing authorization.
Our company is developing a new molecular entity for an ultra-rare fatal childhood genetic disorder. Do you know if FDA has allowed for a more streamlined non-clinical toxicology program for these ultra-rare, life-threatening, or serious diseases, and under what conditions?
Right, that's a good question. That question comes up a lot, and the answer is it depends. The precedent is risk versus benefit, and the FDA is going to take a case-by-case approach for both the toxicology and the clinical program. Under some circumstances, the studies could be abbreviated, they could be deferred, or they can actually be omitted, or you can have additional studies that you need to do on occasion. The ICH M3 guidance document allows for flexibility, and many of the divisions where rare drugs are being INDs are in those divisions. Most of the divisions allow that flexibility. There's a couple of examples. CARC studies may be deferred to initiate at the time of filing. They still have to do them, but they don't have to do them. They don't have to have them completed before they file their NDA. Also, another example might be I've seen where oftentimes for in order to start a 6-month clinical program, you need to have at least 6 months of tox data to support that, general tox data. There are examples where they will let you start your 6-month clinical program with 3 months tox data, as long as the 6-month data is available before you exceed 3 months in patients. And really, it's I think it's important to know that really the rarity of the disease generally does not influence the type of safety studies that are needed. Even for severe life-threatening illnesses, the focus is really to ensure safety. And this includes reviewing all of the animal data, available animal data that you have prior to going into your first in human clinical trials. So I guess it's sort of it's a case-by-case basis, and you really need to provide a strong rationale, and you really need to think about risk versus benefit. But there are several examples where the programs can be streamlined some.
Dana, this is kind of our last question, but it's really more of a clarification that I think the audience wants you to comment on. And the statement is to clarify, more than one pre-IND meeting is allowed if there's a change in who holds the rights to a given technology after the first pre-IND meeting.
Yeah, so I don't think that it's necessarily allowed. I've seen it happen. So is it allowed? Typically, no, it's not allowed. But in this one particular instance, it happened. It was allowed for that one particular instance with this one particular division. They don't have to do that, even if it changes sponsors in between. You can ask for it. They may grant you in another Type B meeting, but not always. They don't have to.
Okay, I think that wraps up our Q&A. There are several questions that you weren't able to get to given time that I know you'll be getting back to those audience members.
Mm-hmm.
I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many great questions. Again, I'd like to thank our speaker for participating and Ro for presenting. A reminder, the webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Again, thank you for joining, and we look forward to seeing you at future events.